either it's endergonic or exergonic, okay? Exergonic, it means that if the reaction takes place, then energy is given off. If that energy is not captured and used in some way to do work, it is lost to the system as heat, okay? Plain. Endergonic requires an input of energy, okay, because it will not be spontaneous. Everything is moving toward low energy states and randomness, okay? That defines the direction that things would proceed. Now, cellular respiration, because I'm going to be going into cellular respiration in the next lecture, it is very exergonic, okay? Minus 86 kilocalories per mole. And I did ask you to know that number, because on quizzes I will not give it, okay? But you'll have to work with it, all right? And I want to emphasize that how this delta G, free energy change, is defined. It doesn't matter how many steps or the pathway by which it's achieved. We're simply taking the end state minus the initial state. And it doesn't matter how it got there, all right? And therefore, standard free energy changes are always the same, always, okay? Now, the actual energy, or free energy free energy changes in a cell is not the standard ones. This is what is done in the laboratory, okay? Because remember the standard conditions of reactants and products starting out at 1 molar concentration. Concentrations of things in cells are on the order of nanomoles, picomoles, okay? Not molar, okay? They're very low concentrations. So, but nonetheless, the laboratory values still give us some insight as to what is happening inside the cells. All right, so I went through all this. We have work. All of these things that I put up there are processes that are and so living things do push endergonic reactions by energy coupling, okay? And so this is linking together an exergonic reaction that is giving off free energy, and is linked to an endergonic reaction that requires free energy in order for it to go. And therefore, usually, it doesn't have to be this, it could be anything any reaction that gives off free energy. But in most cases in cells, it is the hydrolysis of ATP, okay? And once again, I wanted you under standard conditions to know that the hydrolysis of ATP minus 7.3 kilocalories per mole. That's once again another number that I will not give you. For any other things, I will, right? And the value of that hydrolysis applies to the hydrolysis of the terminal phosphate to make a DP, and even the next phosphate to make a MP, okay, but not to the third phosphate. So we could use ATP twice, or we could use 2 ATPs once, and the energy given off, free energy given off by the reaction is exactly the same, right? Okay, so I gave you this example, endergonic, exergonic, and one of them required 3.4 kilocalories per mole, the other one hydrolysis of ATP minus 7.3, and therefore the overall reaction, the overall free energy change of the coupled reactions is the sum of those two numbers. Keep in track what is positive, what is negative, okay? It isn't just the numbers, it's also the sign. Sign is important. And so this here, so it took a little bit of energy here, but basically 3.4 kilocalories per mole to drive this, but we have energy left over, okay? Minus, that is simply lost as heat. And in fact, the heat is what is driving, okay? Okay, because that release of heat is increasing entropy, okay? All right? All right, so now, typical cells at rest. Yes?
Is PI
Phosphate, inorganic phosphate. Inorganic phosphate, okay? Yes?
I thought that, the energy loss, like, from the second to the first one is like less than the last one.
Say what? 7.3, next one's 7.3, this one here is less than that. Yeah, the reason for that is because you have a phosphate attached to a carbon. Okay, here you have oxygen, phosphate, oxygen, phosphate, and a bunch of oxygens, and they're pushing away from each other, the oxygens, because we have two negative charges on those phosphates. Therefore, to hold those together takes more energy, so the bond has more energy. And I didn't give a value for the last one, okay, because we're not going to worry about that. Typical cell, 10 to the 7th power ATP used per cell per second, okay, it's quite a bit. That's a resting cell. If it was a muscle cell that is undergoing strenuous exercise, this could increase by 3 orders of magnitude. Okay, so you can use a lot of ATP, and in order to stay alive, that same amount has to be regenerated. And so if we're using 10 to the 7th power ATP per second, the cell has to generate and create ATP at a rate of 10 to the 7th power per second. All right, and the regeneration of ATP, what do I mean by the regeneration of ATP? We have ADP plus inorganic phosphate to become ATP, okay? And that is intergonic. It requires energy. It's not free. And this is the whole purpose of cellular respiration, to regenerate ATP. And so if end value is to, under standard conditions, to regenerate ATP is positive 7.3 kilocalories per mole, just the opposite of the exergonic reaction. Same magnitude, but opposite sign, okay? Same magnitude, but opposite sign, okay? Now, the reaction is spontaneous if it has a negative free energy chain, okay? That's the important thing to remember, okay? And indeed, that's energetically favorable, and I gave this value for cellular respiration. It has a very high negative free energy change of minus 686 kilocalories per mole. However, if we, this is starting with glucose, okay? Now, if we took glucose, a mole of glucose, put it out on the table, let it freely interact with atmospheric oxygen, and we beat away all the ants and everything else that could possibly interfere with that experiment. This, even though it's highly exergonic, it would take years for those reactions to happen, all right? So being spontaneous says nothing about how fast it happens, okay? That's a matter of kinetics, rate, okay? Here we're talking about thermodynamics. Thermodynamics just only shows which direction it would go if there were a pathway that would allow it to go that way, okay? Kinetics defines how quickly those reactions happen. And so that's real important. So we look at an exergonic reaction. This here axis is, you've probably seen graphs like this somewhat in chemistry, but this here was enthalpy, okay? Now this is free energy, okay? Different concept here, free energy. Delta G is from the energy content of the reactants, or the products, energy content of the products, minus the energy content of the reactants. This is delta G, okay? The ending minus the beginning, okay? Ending minus the beginning, negative sign, okay? That makes it exergonic. However, in order for these reactants to work together, to have any sort of reaction, we have to have things collide. They have to collide, and they have to collide at a very high kinetic energy. They have to have sufficient speed, and also they have to collide at the correct angle, or else they simply bounce off each other, okay? So at absolute 0 Kelvin, things are not moving, and therefore you won't get any reactions. The lower the temperature, the slower the kinetic energy, the less likely things are going to react, all right? So we have to have that type of collision. And therefore the only molecules, even if we have a large population of these things, The only molecules that will reactants that will undergo reaction are the ones that have sufficient kinetic energy, okay? Otherwise, they just bounce off of each other. It'll work. Okay, so I'm gonna jump here to 2 slides, okay, to 26, okay? When you have any population of any substance, it pretty much follows a bell-shaped curve with regard to kinetic energy, okay? So here on the x-axis, the kinetic energy, this is high kinetic energy, this is low kinetic energy. This is the number of molecules in that particular, that have that particular kinetic energy, okay? So in many cases, they can't react, all right? Oh, let me go back, I forgot to say something here. That's important for what I'm trying to do here. Let's jump back to this. Now notice here that there is this hump, okay? That hump is called activation energy, okay? It's not delta G, it's not the free energy. The free energy is from here to here, okay? But a lot of reactions, they have a barrier, okay? This is sort of like having a ball, top of a hill, right? But we have a little hump there. We have a ball, all right? This is what's illustrated in the textbook. But with gravity pulling it down, this would, if it could get over that hump, it would automatically roll down the hill, right? Okay? But this here represents a barrier. It has to get over that barrier, all right? And a lot of times the barrier is having, in chemical reactions, is having sufficient kinetic energy to collide, all right? So that's a barrier. That's called activation energy, all right? So if I go back to slide 23, okay, that's what I have here, E of A. It's the activation energy. And so that defines the kinetic energy at which 2 molecules have to be moving in order to react, all right? So in this population, most of the molecules don't have that high kinetic energy. Just a small little fraction, okay? Only those, this little piece right here, okay? Those are the only ones that have sufficient kinetic energy in order to react. In chemistry lab, what do you do to speed up a reaction? You what? Well, forget about the calculus, what else? You increase the temperature, okay? You boil it or something. Okay, so what's that doing to this population if you add heat to it? It's increasing the kinetic energy, all right? It's going to move the population over to the right. Now we have a larger proportion of the population that has sufficient kinetic energy to undergo a reaction, all right? So speeding it up the kinetic energy, we have more collisions. We've shifted the population over to the right. In living things, to a certain extent, a very limited extent, you can do this, but it's very limited because what happens when you increase temperature? To biological molecules, especially proteins. You're trying to denature them, all right? Now, certain animals that are, and even plants, okay, plants don't have, they have limited control to control their, limited mechanisms to control their temperature. So when I was talking about membranes on the last quiz, giving 3 ways in which you could speed up the glycosity of something that's cold, plants can control temperature, high temperature, lower it by evaporation, but that's very limited. And furthermore, if it's cold, they have no mechanism for warming up, right? It doesn't work. We stay at a constant body temperature, but what happens when you get real sick with the flu? You have a fever, okay? So that has functionality in your body because it is speeding up chemical reactions that would fight the infection. But how high can your temperature go? Not very high, okay? Not very high at all. And if it stays high for too long, it's a bad deal, all right? So we can't do that. So in most biological cases, we're very, very limited as far as speeding up chemical reactions by heating. So there's something else that happens, okay? Something else that happens is that we use biological catalysts. Now we're in your territory, okay? So catalysts, we have to have things happen very rapidly. They can't happen like my glucose here. It would take a couple years for it to completely react. We need to have that happening in a few seconds, right? And therefore, in biology, most of these reactions are catalyzed by enzymes, okay? Most of the catalysts are enzymes. Most of the enzymes are proteins. But some principles here, we don't need a whole lot of them. Okay, they don't have to be in massive amounts. So just a little dab will do you. Okay, just a little bit of enzymes will work. And furthermore, they're not consumed in the reaction. They participate in the reaction. They may undergo, or in fact they do undergo conformational changes, but it's a cycle of conformational changes that at the end of the reaction, they're back in the same conformation that they started with. They do not affect the free energy. They do not change the free energy. Free energy stays the same. What they do, however, is that they provide an alternate pathway that has a lower activation energy than the uncatalyzed reaction. Now, your textbook will say this differently, which I don't like because it's misleading. Okay, your textbook and other things will say that what the catalyst does is they lower the activation energy. That's not true. They provide another pathway for the reactants to proceed, a pathway that has a lower activation energy. Okay, the original uncatalyzed pathway still has the same activation energy. So, all right, and they do this in very many ways. Furthermore, the enzyme, like transport proteins, this is going to be a common thing we see with proteins all over the place, is that when they interact with something else, they have a very specific choice or taste. So we call these reactants a substrate. So when we're talking about enzymes, we're referring to the enzyme and its substrate. The substrate is the molecule that binds to the enzyme, okay? And they're very, very specific, okay? All right, so what happens, we can't do this, right? That just doesn't work. So instead, this is what happens. If we go back to this original case, the activation energy is we have the population, we can't heat it up, we can't move it to the right, but what we can do if we add a catalyst is that we provide a second pathway with a different activation energy, which is lower. And therefore now we have more that can undergo this reaction, okay? And so this is what's going on, all right? So the catalyst, go back to this. So this is the activation energy barrier without a catalyst, without an enzyme. When we add an enzyme in here, it lowers or it provides a second pathway that has a lower activation energy and more of the substrates can react, all right? Okay, all right. Now, this activation energy is actually a series of events that are happening very, very rapidly. For example, we have the enzyme, the substrate binds, and now we have an enzyme substrate complex. And in fact, we have a series of enzyme substrate complexes, each of which have different activation energy levels. And that eventually turns into an enzyme product complex. And then at the end of the reaction, the product is released. Our enzyme is back in the same conformation that it began with, okay? So it goes through these changes of states, and it goes again and again and again and again, okay? So the enzyme keeps being used. And so now where the substrate binds is a pocket or groove on the surface of the enzyme that is called the active site. So that's the binding site for the substrate, but it's given a special name, the active site, all right? So it's always on the surface, okay? It's always on the surface of the protein, and usually consists of just a few amino acids. R grooves, okay? The important thing there is the R grooves, not the backbone. And so this is where the substrate has to fit in, and once again, we have that shape-counter-shape relationship, okay? You have to have the right shape electronically to fit there, and then once it binds, substrate binds, it leads to a conformational change, okay? So this is, I've been giving the examples of key and lock and key, shape, counter shape. However, that is static. Here, once we have the substrate binding, the active site changes shape, okay? So everything changes shape here, and that's called an induced fit, okay? So, cartoon from or drawing from your book. Here's the active site. This is the whole enzyme. And once the substrate binds, we notice that there is a motion of the whole protein, this conformational change, and also changes in the active site. Okay, so that usually this is now has even a greater fit than when it first came into contact with the active site. All right, so some of the features of active sites: small volume of the protein. Okay, the rest of the protein has other functions. But this here is a small volume, and it is three-dimensional. Okay, it's not two-dimensional, it's three-dimensional, and usually, but not always, the substrate is binding into the active site by weak forces, because we got to let go of it as a product a little bit later. However, there are some occasions, especially with enzymes that have metal ions at their active site, which I'll get to a little bit later, there may be a transient covalent bond, okay, but it's very transient. But most of the time, we're talking about ionic bonds, hydrophobic interactions, and hydrogen bonding, all right? Transient covalent bonds sometimes, but they're not permanent. So, once again, these are very specific. So we have an enzyme, for example, we have for sucrose. It requires an enzyme to break that hydrolytic bond between the fructose and the glucose. It has to have an enzyme called sucrase, all right? Sucrase will work with sucrose. But if we have lactose and glucose together, it takes a totally different enzyme in order to hydrolyze that. Now, it's a shame that your book is using this example, beta-galactosidase, because that is the enzyme that bacteria use, okay? Our enzyme, we do have an enzyme that will digest lactose, but it's called lactase. It's similar. Okay, lactase. And so, although these are both disaccharides, we have high specificity as to what enzyme can operate on. So they are very specific. Okay, now how do enzymes affect rates? I have an illustration here of a reaction between a ester, okay, this is an ester, and a carboxylic acid, okay? And if we have, because these things will react, and if we don't have any enzyme or catalyst here, they're separate from each other, the reaction, if we have the rate equal to 1, we can increase the probability of collisions, because these two, the ester and the hydroxyl group, have to collide with sufficient kinetic energy. But we can increase the collision rate by linking them together. Okay, so here is our ester, here is our carboxyl group, but they're covalently linked. These are moving, but now a collision between the two is more likely, because they're not just far away from each other and randomly running into each other. They're close, okay? And the enhancement rate is 5 orders of magnitude without the catalyst, or without holding them together. And so that's something that enzymes do. They have 2 substrates, they have a binding site with 2 substrates bind right next to each other, proximity. But we can even increase it even more, because they have to collide with a correct orientation. even if they're proximal to each other, if they don't hit each other at the same right angle, then they won't react. So now here we put in some cyclic bonds here so that we have locked the ester into a position, a very specific orientation with regard to the hydroxyl group, and that increases the reaction rate by 8 orders of magnitude. And so enzymes do something similar like this. It's part of the story. Part of the story. So they may also, with the induced fit, put stress on the bonds. Okay, so when we have the two substrates bind, we have an induced conformational change. It may put stress on those bonds so that they're easier to break. Those are some things that happen. Furthermore, some reactions proceed best under a certain physical environment. So that pocket, that active site, may have inside of it hydrophobic R groups and creating a more hydrophobic environment, or it might be that they have carboxyl groups and or amine groups that change the pH inside of that pocket compared to what it is on the outside. And this here, changing the environment of where the reactions take place, can enhance the reaction rate. And also, these are always that active site has amino acid side chains. That's what's important there. That's what is grabbing onto the substrates and we have the reactions with. Okay, so most of these enzymes are proteins. However, in the last 20 years, we have discovered that RNA can sometimes function catalytically. And I will, in this class, come across some of these RNAs that function as enzymes. When it's an RNA that has the catalytic activity, it's called a ribozyme. And so in here, we'll come across about 2 to 3 different important forms of ribozymes. Okay, but still, most of them are indeed proteins. Proteins are the workhorses of life, all right? DNA stores the information, but the enzymes, they do all the work, all right? So many different types of enzymes, and I'm going to name some classes here. You don't need to know all of them now, but you will, okay, eventually, all right? So this one you should already know. What do kinases do? Phosphorylation. And on the quiz, I asked, what is the name of the group of enzymes that undercatalyze phosphorylation? You know how many right answers I got? Not very many. Okay, so come on. I know there's a lot of information here, but this is not simple diffusion. It doesn't just float into your head. It's active transport. Okay, you really need to put more effort into it. This is very much knowable. It is knowable. I've seen it for 25 years. There's people who pass this class. Okay. There's people who get A's, right? Last year was the biggest group that I've ever given. Well, no, I didn't say I give A's. They learn it, okay? All right? So you can do it. All of you are smart people. After 4 weeks, I can see that you're smart people. All right? Okay? Encouraged. Entertained. Probably more entertained than encouraged. Okay, so kinase, phosphorylation, and it's a big group. There's many different types of phosphorylases. Peroxidase, okay, I've mentioned catalase. There's other types, peroxisomes. They're substrate. They break down hydrogen peroxide and other types of peroxides. Hydrogen peroxide alone is not the only type of peroxide. Isomerases, how do you convert that number 4 hydroxyl group on glucose into galactose? Okay, bonds have to be broken, switched, resealed. Isomerases, that's what they do. Okay, these are types of oxidases. They oxidize. Okay, we'll talk about that a little bit later on. And reductases, they reduce. Usually these two have to act together, and I'll talk about that in the next lecture. All right. When you see this suffix, ase, it means enzyme. Okay. Os SE is sugar, carbohydrate. A's always an enzyme. And unlike the carbohydrates, not all names of carbohydrates end in OSE, but all enzymes end in ASE. Okay, it's universal. Okay, so that suffix means, and a lot of these enzymes are named after their substrate. Okay, so we have lactose and lactase. Okay, sucrose, axon, or sucrase, axon, sucrose. Okay, that's very common. All right, now it is true that the enzymes are really important for speeding up chemical reactions, but even the enzyme actions rates can still be affected by physical conditions. And you'll be, in a couple weeks, doing experiments like what I'm going to propose here in just a moment. Okay, so for one thing, the rate, it can be determined by the substrate concentration or the enzyme concentration. If we change any one of those, we can affect the rate. Okay, so we're going to do an experiment here that we're going to hold the enzyme concentration constant. So whenever I have anything with brackets, okay, that means concentration. We're not changing the enzyme concentration. But what we're going to do is set up several test tubes here, and all of them have the same enzyme concentration. We're going to put a low concentration substrate here, a little bit higher, a little bit higher, a little bit higher. In other words, from each of the tests here, we're changing the substrate concentration by keeping the same number of enzymes. And lo and behold, what we have, this is the reaction rate. This is also called a velocity, okay? Because velocity is the speed at which something happens per unit time. Time is in the rate, okay? So this could be moles per second, it could be micromoles per second, it could be so many molecules per second, and so forth. That's a rate, okay? Time is there. But if we increase the substrate concentration, we do increase the reaction rate. But then it starts to level out, and if we went to infinity, eventually it would be flat. Why do you think that happens? What? We're holding the enzyme concentration steady. There's only so many active sites. Okay, so this is just like transport proteins, all right? So they can be saturated. Okay, I won't go through my Safeway illustration again. I think that should sink into everybody's mind. You'll remember it every time you go to Safeway. Okay. All right. So yeah, at high substrate concentration, we get saturation. Everyone, all the active sites are acting. All the active sites are full. All the active sites are processing the molecule substrate as fast as they can. We can't go any faster. Now, some enzymes are real good. Some enzymes are rather slow. From one enzyme to the next, their efficiency is not the same. And so one way we can look at their efficiency is to do some equations. So this is a velocity, and where it asymptotes out, okay, is called the maximum velocity, Vmax. Okay, and we can determine that experimentally, all right, by if we have an increase here of substrate to infinity. Okay, now we can't do that in the lab, but nonetheless, this is approaching that Vmax, and so we can sort of estimate the maximum velocity from an experiment like this. You're going to be doing this type of experiment, okay? And now, if we okay, so this Vmax, how high it is, it really depends on the efficiency of the enzyme. How well it recognizes its substrate, can bind to it, process it, let it go. Okay, that has to happen. So one measure here that we can have from an experiment like this is to determine what is the turnover. Okay, the turnover is the number of substrate molecules being processed by one enzyme per second. Okay, so from this experiment, well, we know our enzyme concentration because we fixed it. And we can measure Vmax, and so we get Vmax divided by the enzyme concentration, and we get the turnover rate, right? So this turnover might be 1,000 molecules, 1,000 substrate molecules per second per enzyme. That's actually pretty slow. That's pretty slow, all right? Now, so a higher the turnover, the better the enzyme, okay? The higher the turnover, the more efficient the enzyme. There's another constant here that's worthwhile. It's called the Michaelis constant. Okay, so this is how you get from this to the Michaelis constant. Once you have, holding the enzyme concentration steady, and we're increasing the substrate. We've determined Vmax. If we come down 1/2 that value, 1/2 Vmax, strike a line straight over till we hit the curve, then straight down, then we get the Michaelis constant, which is K subscript M, okay? And what is the meaning of this Michaelis constant? At this point, half of the enzyme's active sites are active, filled. The other half, they're empty, okay? So it's 1/2. Now notice, since this is going down to the Michaelis constant to our x-axis, the units is substrate molecules, okay? Substrate molecules, all right? So what is the meaning of all this? Michaelis constant. So it is the rate at which 1/2 of the active sites are filled. 1/2 are not filled, all right? And this can be used as a measure of how well the enzyme is recognizing its substrate and turning it over, all right? So, and also how quickly the enzyme is at converting S to the substrate to product. Therefore, you know, a high Km value is an enzyme that is not as efficient as an enzyme that has a low Km value. Why do you think that is? Why do you think a high enzyme or a high Michaelis constant indicates something less effective than the low Km? Yep.
A high constant means that the rate is happening very quickly, and for it to happen quickly, the bonds can't be strong because it takes longer to break those bonds.
Part of that's right, part of it is not. Okay, but this is what's happening. Yeah, man.
Yeah, well, if you have a high cam, you need more substrate to reach.
Yeah, in order for that Michaelis, half of the substrate active sites to be active, you have to have a higher concentration substrate. Okay? Now, if something requires a low substrate concentration and is recognizing and breaking it down real fast, you know, that's more efficient. Okay? So, something with a high Km indicates that the substrate concentration has to be pretty high for the enzyme to recognize the substrate and process it. Something that is low Km, because remember the Km is in terms of substrate concentration. Okay, substrate concentration. So the lower the substrate concentration is dilute, and yet the enzyme can still grab onto it, and we have half of the active sites working. So that's pretty good. All right, so low Km indicates high efficiency of the enzyme. And all these enzymes, they're different, all right? You don't have to memorize these, and don't worry about K, cat, or this over here ratio. This is what we're looking at. And therefore, you know, something like this 2.5 times 10 to the minus, the low numbers like that, that's a low number. And therefore, this for fumarase, I believe, yeah, fumarase, you don't need to know these names, but this is some enzymes and their Michaelis constants. All right, so what happens if we do the same experiment with the same enzyme, okay, but we're going to double the enzyme concentration. Compared, here's with one concentration, we're going to double the enzyme concentration. Does that change anything? What is our Vmax? Vmax is twice as high now, because we now have twice the number of enzymes. But, what about the Michaelis constant? It's gonna be the same, okay? It's constant, okay? So, if we have 2 times Vmax, then 1/2 of Vmax 2 is gonna be Vmax 1. Okay? And therefore, if we, we're looking at this, swing it over to here to where we hit, because this here now is 1/2 Vmax of the doubled, enzyme concentration. And now it's here, we come here, and we end up in the very same place. So doubling the enzyme concentration doesn't affect the Michaelis constant, right? If we're doing the same enzyme, right? That's what's important here. Okay, now just to give you some ideas how important enzymes are in speeding up reactions, just look at a very important enzyme, carbonic anhydrase. You have that word up there? Yeah, there you are. Carbonic anhydrase. This in the body forms the reaction between, back and forth, carbon dioxide plus water to make carbonic acid, but it goes the other way as well. And so we find a lot of this in the bloodstream, also in red blood cells and so forth. Now, if we have, this reaction will happen automatically, and if we just set the scale as being 1 molecule per second without a catalyst, and we add the carbonic anhydrase, then with the enzyme, this 5 orders of magnitude, okay? 600,000 molecules processed per enzyme. 1 enzyme, okay? So enzymes can really speed up the reactions. They have to, right? And some of these have evolved to be very, very efficient. Okay, so even with the catalyst, still there can be physical conditions that influence, for example, temperature. Okay, how do you think temperature, if we had, we would be doing this, we're going to set here, test tube with set enzyme concentration, set substrate concentration. Same here, same enzyme concentration, substrate, same enzyme concentration, substrate, but we're going to put that at 1 temperature, this at another temperature, this at a different temperature, and let them go. So how do you think temperature will affect catalytic rate of enzymes?
Higher temperature means higher reaction rate because of more kinetic energy, so fast. Oh. Higher temperature means higher collision rate because there's more kinetic energy.
Exactly. So increase temperature, you increase rate up to a point. And any further temperature increase does what? Denatures, okay, our enzyme doesn't work. So yes, on this first part here, it is increasing because we are increasing the temperature, but we're limited in how much we can do that. We'll denature the protein, and as the active site starts to fall apart, then we get a decrease, okay? So in other words, the enzyme has, and all enzymes have, very specific temperature optimum. Now for our body, 37 degrees Celsius is pretty common, okay, but there are other things like plants where the pH, the temperature optimum may not be that, okay? And furthermore, we have some animals that do not maintain constant body temperature, okay, like snakes. They can have a low body temperature, 10 degrees Celsius, get it up to 20, 30, and so forth. And so they have a big range, and it does speed up their enzymes up to a point when you get too hot, 40 degrees, so they start falling apart. Okay, so increased collision rate on the low side and denaturation of the enzyme on that side. Okay, and so this is something we also see with biological catalysts. All right, but also pH. Okay, and see it here, it can be quite variable. All enzymes have a pH optimum, pH at which they are most efficient. And that can differ in different parts of the body. For example, I have here, you'll learn more about these a little bit later, pepsin, trypsin. Both of these are protein digesting enzymes. Okay, they break peptide bonds. So pepsin is found in the stomach, all right, which is highly acidic. Okay, trypsin is found in the small intestine, which is about 7.2, okay, pH. So when your hamburger first comes into stomach, it's acidified and pepsin is released, and then this can work because it's at about a pH of 2 or so. But and that's a protein. Eventually, all those contents in the stomach get shoveled into the small intestine, and from the pancreas, they're pumping in buffers. All right, so that the pH is buffered up to about 7.2 or so to 8. What happens to our action of when we get into small intestine? Is our pepsin going to work? No. It stops. It can only work in the stomach. And incidentally, we recycle. That protein gets digested and you absorb it into your body. Okay. Now, trypsin comes from the pancreas, and it will start digesting here. All right. So these have different pH optimum. Now, about half of the amino acids that your body takes in across the epithelium of the small intestine is actually your own proteins that you digested, okay? Because these things get recycled, all right? The rest of it is what you ate, okay? All right, so all these, the pH can alter side chain relationships, okay? Hydrophobic bonds and so forth, and that's what's changing conformation of the active site. Now, some enzymes are just protein polypeptides. Some of them can be just one polypeptide chain. Others have ordinary structure and have several polypeptides, and that's all. But there are a lot of enzymes that, in order to work, they have to have added to their active site a cofactor. Okay? So these cofactors are non-proteins. They're not protein. Okay? There's some other type of molecule that has to be put at the catalytic site, the active site, in order for that enzyme to work. And so I'm going to give here two different classes of cofactors. And all these are added post-translationally. Okay, now that's going to be a new term. When we get later in the course, we're going to be using translation quite a bit. It is basically the word for protein synthesis. Okay, all the proteins are polypeptides being generated by ribosome, and that's called translation. And so the protein has to be made first, and then the cofactor is added to it after we make the protein, okay, for its action. All right, and so when I say they may bind the active site, in most cases they do. And furthermore, during catalysis, they also will bind to the substrate as well, okay? All right, so some of these are inorganic ions. One of the reasons, in fact, one very big one, zinc. Okay, you have a lot of proteins that require zinc to work, copper to a little bit, and iron. Okay, in fact, our inorganic ions that we require, zinc and iron, happen to be pretty high on the list. Okay, all right. And for example, oxidases. I mentioned oxidases. They do oxidation reactions, which we'll get to later. Most of them require iron, 2+, all right, as a cofactor. Zinc, 2, is for carbonic anhydrase, which I just gave an illustration with. And furthermore, magnesium. Okay, why do you need magnesium in your diet? It's kinases. And what do kinases do? I like that question. Okay. You need to know my questions forward and backward, because I'll go both directions. All right, yeah, kinases, okay? These you already know. Now you need to know that they require magnesium, all right? Okay, magnesium. All right, and then these are metals, all right? So another type of cofactor category are called coenzymes. In this case, they're organic molecules. They're not protein, okay, but they're organic, right? When I say organic, it means carbon skeleton with other things attached to it. And so some of these enzymes, a lot of them, are derived from B vitamins and vitamin C and so forth. In fact, when we get into cellular respiration, we see a lot of coenzymes that are derived from vitamin B, okay? So a lot of them. some of these vitamins are precursors for making, or some of them are the coenzyme. Some of these vitamins are the precursors for making those coenzymes, right? And without them, we have a problem. So cellular respiration, I'll just name a few. Riboflavin, okay, that's a B vitamin. It's a precursor for some coenzyme that you're going to meet and get very much associated with called flavin adenine dinucleotide. Okay, you can call it FAD. Okay, I would call it flavin adenine dinucleotide because I like to show off. All right, so and also niacin. Actually, what we call niacin, there's 3 different types of niacin. Okay, we now know that, and that is necessary for nicotinamide adenine dinucleotide, NAD. Okay, you don't have to know these yet. I won't ask about them Monday. Okay, but you will. Okay, eventually. All right, and then whatever it is. Okay, coenzyme A. I've already mentioned coenzyme A. You don't know much about it, but you will. Okay, panacetic acid. That's better. Okay, I can do. This is the base form. Panacetic acid is the acid form. Okay, all right. So all these are in cellular respiration, and they play a very, very important role. Biotin, directly. Okay, carboxylases. We're going to come across that type of enzyme later. Okay, it's a class, but they require biotin, and the biotin is the cofactor. Okay, in this case. Okay, so coenzymes are added to the active site. Remove them, the enzyme doesn't work. Okay, so once again, I want to emphasize there are proteins that don't have cofactors. Some of them do. Okay, some of them have to have cofactors in order to work. Okay, another issue with enzymes is that their activity can be inhibited by inhibitors. It can be reversible or non-reversible, irreversible, depending on how the inhibitor inhibits, how it binds. If it's by weak bonds, it can be reversible. If it binds by covalent bonds, it leads to a permanent change, and therefore it's irreversible. All right, so reversible weak bonds, some types of very important inhibitors that inhibit a lot of different enzymes is carbon monoxide, okay, hydrogen cyanide, okay, and these are all irreversible, right? Now, carbon monoxide not only inhibits certain important enzymes, it also inhibits hemoglobin's ability to carry oxygen, okay? Why you don't put cyanide in your lemonade and drink it is because what exactly it inhibits is something in cellular respiration called cytochrome oxidase. And we'll come across cytochrome oxidase a little bit later. It is one of the last enzymes in that pathway, but if it's inhibited, you don't regenerate ATP, and you run out of ATP in just a matter of seconds, okay? It's quite fatal. This is a very important enzyme. We'll come back to that later. So, now these inhibitors that can inhibit enzyme action, they can do it in 2 different categories of mechanisms, competitive versus non-competitive. Competitive inhibitors are substrate mimics. So some part resembles the substrate, and it binds to the active site, okay? Now, if this inhibitor is bound at the active site, then the normal substrate can't bind, all right? We knock it out. Now, sometimes the competitive inhibitor can be processed by the enzyme. In other cases, not. But still, even if it's processed, it's preventing our normal substrate, which we need, to go, okay? So something that sort of resembles this is if ethanol is a poison, okay? And the other type, ethanol and what is it, manganol, what is it? 1 carbon. Alcohol. Methanol. Okay. I'm surprised I forgot that. It's Thursday and I'm waiting for the weekend before my brain goes. Okay. So methanol, yes. Okay. It's worse poison. So what do they do to you if you drank methanol? Okay, it gets processed, both ethanol and methanol are processed by the same enzyme. Okay, this is one case where both of those can be processed. Ethanol, when it's processed, is acetaldehyde. Methanol is formaldehyde. Formaldehyde is really, really bad. Okay, so if you drank methanol, what they would do, they would give you a bunch of ethanol, get you drunk. Okay, why? Because the methanol and the ethanol is competing for the active site with the methanol, and it slows down the rate at which formaldehyde is being made. Therefore, your body can get rid of it without having a bunch of poisonous activity. So that's sort of like what I'm talking about here about competitive inhibition. Okay, so this can be overcome by increasing the substrate level. Why would that happen? Let's say we have equal numbers of substrate, equal numbers of competitive inhibitor. Okay, and let's say that the enzyme has equal affinity for both. So half of the time, the active sites are going to be bound by the substrate. Half of the time, they're going to be bound by the inhibitor. Now, if we double the substrate concentration, why would that speed up the reaction? We're doubling. Now we have twice as much substrate as inhibitor. And therefore, the active site 1/3 of the time would be occupied by inhibitor, 2/3 of the time it's going to be occupied by the substrate. Okay? So by increasing the substrate concentration, we can overcome the inhibition in this particular case. All right, so this cartoon showing it, this doesn't quite look like that, but there is enough, this is normal substrate, but it has at least enough similarity to the substrate that it can bind to the active site. Okay, and as I said, it might be processed, it might not. But the whole point is, as long as that inhibitor, competitive inhibitor is there, then it's the normal substrate will never bind. Okay? Non-competitive. Okay, they don't bind at the active site. They bind somewhere else. Okay? Somewhere else where the shape of the protein is just right for that inhibitor to bind. And so what happens when it binds? Not at the active site. Conformational change. And a conformational change that alters the shape of the active site. So it's, now this one here can't be overcome, non-competitive. So here's the active site, or non-competitive inhibitor is bound somewhere else here, leads to conformational change. And now the active site doesn't have the right shape to recognize the normal substrate anymore. Okay? Now this cannot be overcome by adding more substrate, because substrate doesn't play a role. Okay? We have to get rid of the inhibitor. Alright, so this is non-competitive inhibition. Okay, so many things. Cyanide is a non-competitive inhibitor of cytochrome oxidase. It doesn't bind to the active site. It is changing the shape. And lead is high lead in your body. It inhibits non-competitively a lot of important enzymes, not just one. Okay. All right. Okay. So enzyme regulation, changing stories now. Okay. Enzymes are in pathways. Okay. These are metabolic pathways. One reaction, next reaction, next reaction, and we have to be able to turn enzymes off and on. We need to control the rate of the reactions. Once we get enough product, we need to slow it down. Okay, if we need more product, we need to speed it up. And so there's several mechanisms in general how this is done. Some of these are post-translational modifications. I'm going to give two examples. Some enzymes are made in an inactive form, and they have to be modified to be in the active form. Okay, for example, some that require cofactors. Okay, they may not work originally. There's a class of proteins, which includes some enzymes that are called zymogens. These are also in an inactive form, and they have to be cleaved. A specific peptide bond has to be broken, which leads to the conformational change, which changes their action. Okay, the enzymes I gave earlier, pepsin and trypsin. They're made inactive, and they don't become active. Pepsin doesn't become active until it's dumped into the stomach. It is a peptide bond that's broken by the high acid activity, acid hydrolysis, then it's active. Reason for that is if it were active in the cell, it would chew up all the proteins, okay? So it's put out in a inactive form and activated where it needs to work. And that's also true of trypsin as well, okay? But one of the problems with these zymogens, once you get them in the active form, you can't reverse it. The only thing you can do is destroy it, all right? That's the only way to get rid of it. All right, so that's one post-translational modification for regulating the enzyme activity. Another one, which is reversible, is to control the cofactors. For those enzymes that require a metal or a coenzyme, those can be controlled and turned on and off. A more widespread, and some of them do act like that, but another form of regulation is called allosteric regulation. This is really widespread, so I'm going to turn my attention to that. Allosteric regulation, usually these enzymes have quaternary structure, okay? And that could be 2 subunits, could be 3, could be 4, could be 5, could be 6. But what is very often the case is that each subunit has the same active site. So the enzyme has more than one active site for the same substrate, all right? And so more than one active site. And because we're not at absolute zero, these protein conformations are wobbling all over the place, okay? So we can have 2 different forms, the T form, which is inactive, and the active form. So we have these things going back and forth real quickly, okay? Now, we might have an enzyme that most of the time, most of the population's on the active side, but then it'll toggle to the inactive, momentarily jump back, okay? Other classes of enzymes, we might have them mostly in the inactive form, but they toggle momentarily into the active form and back, okay? But we can lock these with allosteric regulation. What happens here is we can lock them into either this form or that form, okay? By having certain substances there. So these enzymes have a special site inside the active site, something called the regulatory site. Okay, this is somewhere on the protein. The active site, or the regulatory site, is also called an allosteric site. So active site, or excuse me, regulatory site, allosteric site, same thing, all right? So here, if we have an allosteric enzyme, and let's say that this one here is mostly in the inactive, okay? So down here is non-functional, inactive, and this is the active form. We have 100 of them in this form, and maybe every second or so, one of them toggles over to here, then it toggles back, okay? So most 99 of them are in this form, and one of them is like this. So if we add a allosteric, well, let's do it the other way around. 100 of them are active. I didn't realize that was coming first. 100 of them are active form, and only one of them in the inactive form. We can turn it off by adding what is called an allosteric inhibitor that binds the allosteric site and locks the protein into the inactive form, okay? Now, first we have just one here, but it's going to toggle back. But once this allosteric inhibitor binds, it's locked here, and then a nanosecond later, we have another one of these toggles over, it gets bound, another one toggles over, it gets bound, another toggles over, it gets bound. And so in a very short period of time, we can take all 100 of these and put it into the inactive form, all right? And to turn it back on again, we just get rid of the inhibitor. Okay, now this inhibitor is an allosteric inhibitor. It's not like the competitive and non-competitive inhibitors I talked about earlier that are poisons, toxins. This is something used by the cell to control their enzymes, all right? The other way, as I said, we can have most of them in the inactive form, and they toggle momentarily into the active form. We can lock them into that active form by having an allosteric activator. The allosteric activator will bind to the allosteric site, lock it in the active form, and therefore we can convert the whole population into the active form. So these are controlled by the cell can control these enzymes by controlling the inhibitors and activators. Okay, sick population. Okay, there's some other feature here of enzyme activity that's sort of similar to this, to allosteric regulation. This is called cooperativity, right? Now, I'll explain the difference here in a moment. Once again, we have enzymes that have quaternary structure, where there's more, each subunit has more than one or each subunit has the identical active site, so we have more than one active site. And these are also toggling back and forth between high affinity and low affinity states, back and forth, but by binding the substrate to the active site, it locks it into the higher affinity form. It's similar to allosteric, okay? But what's happening here is that it's the substrate that locks all of the 4 units, or, well, it's a shame that they use 4 units, 4 subunits, because quaternary structure can be 2, 3, 4, 5, all right? But they're all identical. And the binding of one, so first of all, the affinity for the substrate is relatively low. But once we bind the first substrate there, then all of the active sites are in the high affinity form, okay? It's locked there. So the binding of the first substrate is slow, the binding of the rest of the substrates is fast, okay? This is cooperativity. Now, what's the difference between this and allosteric regulation? Because it's very similar, very similar. Okay, what is controlling, locking the enzyme into the high affinity or low affinity state are inhibitors, allosteric inhibitors, or allosteric activators that bind the regulatory site. Not the active site, not the active site, okay? Something else. Here, it's the binding of the substrate to the active site that locks it into the high affinity form, okay? Any questions on this? Yes?
How does the cell take out the inhibitor and activators? Do you have to put like some sort of energy to like reverse it? Or
Usually, yes, and I'll come back to something in a moment that addresses that, okay? All right. Cooperativity, all right? What are the ways in which we can tell, go back to that experiment that you get to do in a couple of weeks, where we hold the enzyme concentration constant, and we're going to increase the substrate concentration. From the curve here that's produced, we can tell whether it has 1 active site or 2 active sites or more. If it only has 1 active site, that rate change is pretty smooth. If it has 2 or more active sites, the curve is going to be sigmoid shaped. Okay, it's going to have an S shape. It's not going to be a smooth curve. Okay, and so just from this experiment alone, we can tell whether we're dealing with an enzyme with 1 active site or 2 or more. Okay, all right. So that has quaternary structure, has more than 1 active site. Or the enzyme. Okay. All right. What else? Okay. Yeah. This sort of addresses your question. So we have enzymes arranged in metabolic pathways in a very, very specific order. Very specific order. Okay. So isoleucine is an amino acid. Threonine is an amino acid too. But to make isoleucine, it requires this pathway. So the beginning substrate here is threonine. It is converted into one form by enzyme 1. Now, I'm not going to give the names of these. We don't need to worry about that. And into enzyme by enzyme 2, the B, enzyme 3, enzyme 4, enzyme 5. So there's 5 different enzymes, 5 different chemical reactions that have to act in this order. And so once we get enough isoleucine, we want to shut it down. We want to slow the pathway, all right? So what we could do is have an inhibitor inhibit that, but that gets to be complicated, but it's very simple. What usually happens here is that the end product of the pathway is an allosteric inhibitor of some enzyme at the beginning of the pathway, okay? So once we get enough isoleucine, it may bind to the regulatory site on enzyme 1, and it shuts it down. It's conformational, it's in the inactive form. All these others can stay in the active form, it doesn't matter, because enzyme two requires A, and it's not getting A, okay, because enzyme one is shut down, okay? So we don't have to shut down the whole system. Now, this is really common. This is called feedback inhibition. Now, if we then consume the isoleucine, the level gets down and it comes off of this enzyme, and we turn the system back on. So by controlling just the end product, and usually the isoleucine will be consumed, it gets incorporated into polypeptides. And when that goes down, we have this pathway on here. So this is a very common type of thing. So feedback inhibition is when the end product of a pathway is an allosteric inhibitor of some enzyme at the beginning of the pathway. It doesn't have to be the first enzyme, it could be the second, but still something at the beginning pathway. So this is an allosteric inhibitor. Okay, so does that sort of address what you were at? Okay, yeah. Okay, so this is enough on metabolism, and I believe that this will also be the cutoff. I haven't looked at the syllabus, the schedule. Anyone have the schedule here about the first exam? Just to hear? Yeah, so this is the cutoff, okay? So what I say next will be on the second exam, okay? Any questions? Still have some time. So we can get started. So all this sets the stage for photosynthesis, understanding photosynthesis and cellular respiration. Right? Any questions on the first? Well, I've got them. You okay?
Yep.
Okay, all right. All right, so cellular respiration. The energy flow, I showed this before at the beginning of metabolism, and I'm going to expand on it a little bit. One of the things here is energy flows through different things. Eventually, it's all lost as heat. And so what is very important is photosynthesis. We're putting together carbon dioxide and water and forming organic molecules, and that happens in eukaryotes, in those organisms that have chloroplasts. Okay, animals, heterotrophs, don't have chloroplasts. And this gives us organic molecules that are also burned in cellular respiration. We release the bond energy that can be captured in ATP, okay? So this is a more complete diagram of what's going on. Now, notice here that matter gets recycled. Oxygen is an input into cellular respiration. An output is carbon dioxide and water. Carbon dioxide and water are inputs into photosynthesis. So our water and carbon dioxide, they recycle through life. But energy is one-way flow. It comes in here, gets stored here, it gets released here, and even up here. These chemical reactions are not free. You still lose some of the energy as heat. Eventually, all the energy flows through life one direction, okay? It may be passed from one organism to the next organism to the next organism up a food chain, but eventually, all that stored energy is lost as heat, okay? It's not recycled, not recycled. So what happens here, so the matter is recycled, but the energy is not, right? So how this works in cellular respiration is we start with complex organic molecules and break them down to simple molecules. And this is a pathway, okay? A catabolic pathway, degradative, releases energy, all right? So keep straight exergonic, endergonic versus catabolic and anabolic. They look similar, but they are different. When we're talking about exergonic, endergonic, we're talking about individual chemical reactions. Here, anabolic, catabolic, we're talking about pathway, many reactions. Some of which could be endergonic, some of which could be exergonic, but the whole pathway is catabolic, okay? So this energy is released, and that energy, usually, it's abundant energy, it's too destructive, and therefore it is captured in a smaller molecule in incremental steps, ATP. Then ATP can, in that process, some of that energy, in fact, a good part of it is lost as heat. So I'd like you to do this on your own. If all of the energy for cellular respiration of glucose, complete oxidation of glucose, carbon dioxide and water, under standard conditions, 1 mole, okay, minus 686 kilocalories per mole, how many ATP could you regenerate? You'll find, I'll leave it up to you, figure out how many. You'll see that in cellular respiration, it's far less than that, because a lot of this energy is just simply lost to heat. Okay, all right, so now you have to memorize this. Okay, I've showed it before, now you have to know it, all right? So once again, 1 mole of glucose combines with 6 moles of oxygen, 6 moles of water, produce 6 moles of carbon dioxide, 12 moles of water. And I want the water on both sides because this is a reactant, that's a product, and they come about in totally different places within the process. And so, under standard conditions, we have 86 kilocalories per mole. We capture so many ATP, but most of that energy that's released is lost as heat. Okay, it's not totally efficient. So, and the whole function is to regenerate this. Cellular respiration is to regenerate ATP. All right, and as I said, this is positive 7.3. So how would you figure out how many ATP if you could capture it all? You divide this by that. Anyone have a calculator?
94, 93.
What is it? How many? What? 93.3. You'll find that it's about a third of that. Is that, okay? Come back to it later. Okay, so this is not a single step. It could be an explosive single step, it could be too problematic. Most of these reactions are oxidation reduction reactions, okay? And they occur together, so we call them redox reactions. So I'm going to review, how many of you covered oxidation reduction in chemistry? Because I don't know if it's in chemistry 1 or 2, A or B. Okay, okay. All right, so I'm just going to review some of this. So, because it's going to be a little bit different here in a biological system, I'm going to make it a lot simpler than it is in chemistry, okay? So, this is a partial or complete transfer of electrons, okay? That's an oxidation. Okay, so oxidation is a partial or complete loss of electrons. You probably have heard it's complete loss of electrons. I'm coming back to that partial in a moment here. All right, and reduction is a partial or complete gain of electrons. Okay, so these have to happen together, all right? So one reactant is oxidized, the other one is reduced, okay? And I'm going to point out here too that oxidation involves a decrease in free energy, and reduction is a gain of free energy, okay? So if we have this equation here, the reaction of methane to 2 oxygens to produce carbon dioxide and 2 water, and we release energy. Which one of these 2 are being oxidized and which one are being reduced? Okay, first of all, we look at the charge on that carbon atom. Does it have any partial charge? No, it has a charge of 0. But if we strip off the hydrogens and attach oxygens, is there a partial charge on that same carbon? Yeah. And what is it? Partial positive. This here is 0 to partial positive. That is a loss of electron density. Okay, so a reduction or an oxidation is not just only the complete loss of an electron or complete gain. It's also where those electrons are with regard to the reference atom. Are they drawn closer or further away? Okay, so in chemistry they probably give you oxidation states, so we're going to worry about that. I'll come back to that. So it is a methane that's being oxidized, and it's the water that's being reduced. Because does this oxygen here, do they have a charge distribution? equally electronegative, but what about here? The oxygen, positive or negative? Negative, that's a gain of electron, okay, partial gain. It gains charge, okay? And therefore, that is what is being reduced. Okay, so whenever I give an equation like this, and I ask you to tell me what is being oxidized and what is being reduced, don't include the products, because it's the reactants that become oxidized and reduced, not the products. Okay, that's in state. All right, and now, so let's go back here to cellular respiration. This equation, what of the reactants is being reduced? Talk about carbon. Carbon to water, okay? So that's oxidized, actually to carbon dioxide. Eventually, now we do have oxygen in several places here, but the carbon is becoming, and this molecule is ending up as carbon dioxide. And so that's oxidation. Okay, now many of these carbons, they do have a partial negative, a partial positive charge, but not as great as here. Okay, so it does represent a oxidation. And what is being reduced is the oxygen. The oxygen becomes water. Okay, and so that's a reduction. Now, confusing, but I'm going to give you something simple to be able to tell. All these electrons are being transferred between molecules by moving hydrogen. So another way we can talk about this, because you can easily see the hydrogens, even though you can't quite grasp the oxidation state, still an oxidation would be removing hydrogen. That's oxidation. Gaining hydrogen, adding hydrogen, is a reduction. And so if we go back here, this here carbon, we have hydrogens attached to it, but when it's over here, there's no hydrogen. We removed hydrogens. That's an oxidation, okay? And likewise here, we have oxygen. It is reduced because now we've attached water to it, or hydrogen to it, or water. That is a reduction, okay? And it always involves the reactants, okay? So this makes it easy to see what's going on, all right? So, now, this energy that's released